Welcome everyone to our very first lecture in medical microbiology. In this session, we'll build the foundation for the rest of the course. We'll discuss what microbiology is, a brief history of how it developed, and why it is so crucial in the practice of medicine. By the end of this lecture, you'll understand how microbiology connects to diagnosis, treatment, prevention, and medical research. What is microbiology? Definition. Microbiology is the study of microorganisms, tiny living organisms that cannot be seen with the naked eye. These include bacteria, viruses, fungi, parasites, and prions. Some of them are harmless or even beneficial, while others cause serious diseases. When you think of microbiology, don't just think of germs that make people sick. Think also of gut bacteria that help us digest food, or yeast that helps make bread and medicines. But in medicine, our main focus is on the disease-causing organisms, the pathogens. Without microbiology, we wouldn't know how bacteria cause pneumonia, or how viruses like influenza spread. History highlights. Anton van Leeuwenhoek, 1670s, first person to observe microorganisms using a simple microscope. Louis Pasteur, 1800s, father of the germ theory of disease, showed that microbes cause disease and developed vaccines. Robert Koch, 1800s, proved that specific microbes cause specific diseases. Right-pointing arrow. Koch's postulates. These names may seem like history, but their contributions shape everything we do in modern labs and hospitals. Pasteur showed us that diseases are not caused by bad air or miasma, but by microbes. Koch gave us the scientific method to prove which microbe causes which disease, for example, Mycobacterium tuberculosis for TB. Why microbiology is important in medicine. 1. Diagnosis. Identifying the microorganism causing disease. Example. Diagnosing pneumonia. Right-pointing arrow. Is it bacterial, strep pneumoniae, or viral, influenza? 2. Treatment. Selecting the correct antibiotic or antiviral. Example. Giving penicillin for bacterial infections, not for viral flu. 3. Prevention. Vaccination, sterilization, infection control. Example. Hepatitis B vaccine prevents chronic infection. 4. Research. Discovering new drugs, vaccines, and diagnostic methods. Example. PCR revolutionized COVID-19 diagnosis. Microbiology is not just about memorizing names of bugs. It's about applying this knowledge in real life. When a patient comes with fever, cough, or diarrhea, we use microbiology to decide what tests to run, what treatment to give, and how to prevent spread. Real-life applications. COVID-19. Identified and diagnosed rapidly with PCR. Right-pointing arrow. Global example of microbiology in action. Tuberculosis, TB, Ziehl-Neelsen staining in labs helps confirm diagnosis. Hospital infections. Hand hygiene and sterilization protocols come directly from microbiology research. Think about the last pandemic, COVID-19. Within weeks, scientists sequenced the virus, developed diagnostic kits, and within months, vaccines were available. This was only possible because of decades of microbiology research. Another example is tuberculosis, still one of the leading causes of death worldwide, and diagnosis relies directly on microbiological staining and culture. Closing and next lecture preview. Asterisk. So, to summarize today's lecture, microbiology is the study of microorganisms, many of which are responsible for human disease. It began with pioneers like Leeuwenhoek, Pasteur, and Koch, and today it remains central to medicine, from diagnosing infections to developing vaccines and controlling epidemics. In our next lecture, we will explore the different types of microorganisms, bacteria, viruses, fungi, parasites, and prions, and see how each of them behaves differently in the human body. That's a wrap. Don't forget to like, subscribe, and hit the notification bell for more educational content. If you have any questions or topics you'd like me to cover next, let me know in the comments.